Hey guys, welcome to Cosmic Solutions, Dr. Priyanka's Biology, where we discuss things from biology to biotechnology. We had already discussed about transformation. Okay, so we had seen what is transformation, what are the methods, and the end result is such that the transformant what we have, it should have a recombinant DNA. That is what is going to actually define the success of your transformation. So today's session we will discuss on what are the methods for the selection and screening of recombinant transformant. So let's get started with the today's session. Yes. So we will consider three situations. Suppose now we have a transformation. Successful transformation has occurred. Okay. Now this is one E. coli cell with its host DNA. Then we have the plasmids. Now these plasmids, they have, okay, during recombination, they have undergone self-ligation. Okay. And then we have the plasmids which have undergone recombination. Now after transformation, there are three possibilities which can occur. One is okay, first condition is such that okay, no foreign DNA has entered inside the given cell. That means that is non-transformant. And now the remaining two, what the situation is such that one of the cells they have taken up the non-recombinants, whereas the other one has taken up the recombinant DNA. Okay. So the first condition is not what we are interested in it because it forms the non-transformant category and we are looking for this situation, that is transformation. Okay, where transformants have formed. Now, out of this, the first one what we have is it is non-recombinant transformant. Non-recombinant transformant. And this situation what we have is the recombinant transformants. Now whatever we are aiming for, it will be present in this situation. That is the recombinant transformant. And we are more interested in screening of these recombinant transformants. Okay. Now suppose consider this culture plate. Okay, if you can see this culture plate, there are two types of dots here, red and the blue ones. The red dots indicate the recombinant DNAs, okay, recombinant transformants, whereas we have the blue ones which indicate the self-ligated vectors indicating the non-recombinant transformants. Okay. Now we need to identify the recombinant transformants from the non-recombinants. So how do we go about that? So we need some sort of strategy because at the end of the day we want the recombinant transformants because that is what is going to give us the product which we are looking for which we have carried out the entire process of transformation. Alright. So now let's get started with this part. Alright. Now let us take one situation wherein now we have say around 1 nanogram of plasmid, because when we are talking about genetic engineering and all, definitely we will be talking in very minute quantities at the nano micro level, because I am expecting most of this crowd who is listening to this will be from the biotechnology or the life science background because we are talking in detail about this process. Okay. Now the plasmid what we have, suppose around 1 nanogram of plasmid we have. Okay. Now out of this plasmid is capable of yielding somewhere around 1000 to 10,000 transformants. Like overall. Okay. Out of which some will be recombinant whereas some will be non-recombinant. Okay. So if we consider the success of transformation, it is just around 0.01%. Okay, that means very less amount of plasmid is available. Now, so very small proportion of the total number of cell, okay, which is present in that competent culture can give rise to transformer. So the success rate, if you see, it's bit lower. Okay, so now from this smaller proportion, we need to look for some things which are now transformants, sorry, recombinant transformants. So we need to have some method which will help us distinguish between the types of cells. So we have two things, selecting the transformants and non-transformants, and among the transformants we need to select the recombinant transformants and the non-recombinant transformants. Now it's not just that, but for a successful experiment, it is not some demo. Okay, when we are going to carry out such experiments for the research work, it will not be for some demo, but we will do it with some sort of intention in order to express some gene. So here it is very important that there is successful transformation of the recombinant as well as there is stable retention of these plasmids. Okay, so we are looking for stable retention and good expression of the genes carried by the plasmids. So that is what we are looking for. Now let us take the example of PBR 322. So that's a plasmid that we have studied when we were discussing about the uh, vectors. Now if you see this PBR 322 plasmid, okay, there are different components here. Okay. Now, very important is origin of replication. Then apart from this, we can see there are different multiple sites. Okay, PST1, EcoR1, HIND3, EcoR5, BAMH1, CEL1, then NDE1. So like this, we have so many sites. Okay. Then apart from this, we have the ampicillin resistance gene and the tetracycline resistance gene. Okay. So within these genes, we have the restriction sites. Okay. So this is one ideal one which is normally used for variety of experiments. Now, its beauty is that it is resistant. Okay. E. coli cells which will take up this plasmid, that is PBR 322, it will have resistance to both the types of antibiotics, that is the ampicillin as well as the tetracycline. Okay, which otherwise are sensitive. Okay, the antibiotic is going to kill if they do not have this plasmid. Now, because now your PBR 322, it has two sets of gene. Okay, now we have one gene which is present, that is beta lactamase enzyme. Now this beta lactamase enzyme, what it does is, okay, it has the ability to modify the ampicillin in such a way that it becomes non-toxic. Now suppose now your cell, okay, E. coli cell has taken up, this E. coli cell has taken up the PBR 322 plasmid. That means it is going to have the ampicillin resistance as well as tetracycline resistance. Now how this resistance actually works? Now this is because of two sets of genes. Now one gene, what is called as beta lactamase. Okay, it is beta lactamase. Okay, beta lactamase. What it does is it modifies this ampicillin in such a way that it makes it neutral. It is no longer going to harm the bacteria. So suppose now we have ampicillin present in the nutrient medium. So because of the activity of your PBR 322, the gene beta lactamase present, it is going to nullify the effect of ampicillin which is present in the medium. Okay, that is one. Second one, how it works, the second set of gene is the gene that codes for the enzyme that detoxify the tetracycline. So this is how actually your PBR 322 is going to work. Point 1 is because of the action of beta lactamase enzyme which is going to modify or make ampicillin non-toxic, and second one is the detoxification of the tetracycline. So this is how your PBr 322 work. Now suppose now we have carried out successful transformation using PBr 322 as our vector. Okay. Suppose now we have used this particular enzyme for our work. Okay. The E. coli, okay, so only those E. coli which have taken up, okay, the plasmid, successfully they have taken up the plasmid, they will be ampicillin resistant and tetracycline resistant, right? Rest all they will be sensitive because by default they do not have the resistant genes, okay. So they will get the resistance of your ampicillin as well as tetracycline. So others they will die. So definitely here the non-transformers, because they will not have the plasmid, hence they are ampicillin sensitive and tetracycline sensitive. So when we have the culture medium containing the two antibiotics, that is your ampicillin and the tetracycline, the sensitive ones they will definitely die. That means your non-transformants are not going to grow. So the question of selection of your transformants and non-transformants is over there, because we have only the transformants. Now the next problem what we have is the selection of the recombinants and the non-recombinants, because transformants can be both. That is the ones which are carrying the recombinant DNA as well as the non-recombinant DNA. So the main question is the selection of your recombinants. Fine. So identification of the recombinants. Okay. So how do we go about it? So most of the cloning vectors, insertion of DNA into a fragment of plasmid normally destroys the functionality or the integrity of the genes present on the molecule. Now this is what we had already discussed in one of the sessions before, that suppose now, okay, let's take some different color. Okay, now here we have this gene which is giving the ampicillin resistance, and here we have a gene which is giving the tetracycline resistance. Suppose now we choose BAMH1 as the site where your restriction enzyme is going to make a cut or nick. That means it is going to break the sequence of this gene which is responsible for your tetracycline resistance. So it is going to break it. Fine. So it is no longer functional. Okay. Now, nowadays we use the phone. Okay, your smartphone for the communication. So only when your phone is intact, okay, it is having the battery, SIM card, everything, it is going to pass on the message, or you can carry out the function what you are expecting. But if we are doing some nonsense with our phone, that is if we are removing the SIM card, or if we are making some changes in the SIM card, it is no longer going to be functional. So similar thing is happening. So we are breaking the integrity of any one of these genes, so that it is no longer going to give the same message which it was supposed to do. So that is done by inactivating it. Okay. Fine. So such a method by which, okay, the functional gene is inactivated by incorporating or inserting the gene of interest within the sequence of the gene present in the plasmid. It's called as insertional inactivation. So this is one of the methods that is used for identifying the recombinants from the non-recombinants. Okay, so apart from insertional inactivation, we have some more methods. Okay, so one of them is the insertional inactivation, as we have discussed. Then we have something called as the antibiotic sensitivity. So far whatever we have studied or whatever we have discussed about the activity of the antibiotics or the antibiotic resistance gene present within the vector or the plasmid, that is what we were talking about, the antibiotic sensitivity. That was the first method. Second method is the insertional inactivation. Suppose now I have this pen. Okay. If now it is working because it is intact, okay, it has its refill intact, its nib is intact. Suppose now I break this or I cut this pen and I insert something else inside it. It is no longer it is going to be functional, right? Because its main integrity is broken. Okay. So now insertional inactivation normally works in association with antibiotic sensitivity. So these two methods, usually all three methods, they go hand in hand. So this antibiotic sensitivity is there for it works together with other methods. Okay. So usually the selection is done with the help of these two. And the last method which we will study further is the blue-white screening, which also we have already discussed. Alright. So coming back to insertional inactivation, there are two methods of insertional inactivation. Okay. Now going back to this PBR 322. Now as we had seen in this picture, okay, the previous picture, that your PBR 322, it has so many restriction sites. Okay, so let me clear this first. Alright. So it has so many restriction sites as we had seen. Alright. And these can be used to open up the vector before insertion of the new DNA fragment. Okay. Now, as we had taken the example before, BAMH1. Okay. So, it is present within the gene which is coding for resistance for your tetracycline. Okay. It is present over here. Okay. It is present in the gene which is encoding the tetracycline resistance gene. So, if we carry out the cutting or the nick is being produced with the help of this enzyme, okay, restriction enzyme at the BAMH1 site, it is going to break the tetracycline resistance gene. Okay. So, now the recombinant PBR 322 what we have, it will have one additional piece in the BMH1 site. So it is no longer going to be tetracycline resistant. So it has lost its ability to give resistance to tetracycline. But we still have the ampicillin resistance gene intact. Though this is gone, but we still have our savior, that is the ampicillin resistant gene. Suppose if we make such a recombinant and if we want to select it from the non-recombinant transformants, what we would do is we will add ampicillin. We will add ampicillin to the culture media. Okay, we will add ampicillin to the culture media, and what will happen is because of this ampicillin resistant gene, intact ampicillin resistant gene, only those cells, okay, only those transformants which have this broken tetracycline resistance gene, that means the cells or the plasmids with the new foreign DNA, okay, they will only those ones will grow. So that means because of the ampicillin resistance gene, we will be able to identify the colonies. Okay. So this is where your insertional inactivation has come into picture. So next one. So how to detect the cells which contain the recombinant DNA. So we have already seen that. So whereas the non-transformants, because they will be sensitive to ampicillin by default, they will not grow. So the question of non-transformant and transformant will not come into picture because the non-transformants will only be ampicillin sensitive. Alright. So the next method is the replica plating. Okay, so the first method of identification of your Recombinant was your insertional inactivation. So second method what we have is the replica plating. So when we talk about replica, that means identical. Okay. So normally, like those of you who are looking like your sisters or those of you who are looking exactly like your mother. So people must be normally saying, okay, you are exact replica of your mother or you are exact replica of your sister. That means you look the same, mirror images. Okay. So we have to use this method of replica plating. So plating is basically we are going to produce an image of the original one. Okay. So how that is done? So we'll see now. Alright. So what is done is to identify the recombinants, the colonies are replica plated onto agar medium. So now we have one, now we have carried out transformation, right? Recombination is done, transformation is done. So here we know that, okay, your cells what we have, okay, let's take this another slide. So yes. And let us take this slide. Now we have this one culture plate. Okay. So suppose now we have the transformants here. So some are recombinant, some are non-recombinant. Okay. So what we will do the replica plating. So in replica plating, what we shall do is, okay, we will have agar medium which will contain the tetracycline. Okay. Agar medium, we will have one plate, okay, which will have tetracycline in it. We have added tetracycline to the medium, growth medium here. Okay. Now what we will do, this original plate what we have, okay, we are going to, okay, do the pressing over it so that the impressions are being produced. So when I am talking about the impression, basically I am bringing the colonies from your previous plate, okay, this plate, in contact with this new fresh plate which has tetracycline added to it. Okay. So when it is touching, definitely we are making sure that the new colonies have to grow. Okay. So after replica plating, we are going to incubate the plates and we will wait for the colonies to grow. Now here what will happen? Only those will grow, okay, which consists the cells that carry the normal PBR 322 and not with the inserted DNA. So why? Why this is happening? See, we have to consider two things here. Now here on this place we have tetracycline resistance. Now suppose now recombinant ones what we have. Here your tetracycline is broken because the gene has been inserted into the tetracycline resistant gene, right? Whereas here your tetracycline will be functional. In case of your non-recombinant ones, tetracycline will be intact, right? That means it is those cells they are going to give the tetracycline resistance. So when we grow, okay, when we carry out replica plating and allow to grow, okay, on the tetracycline containing plate, so what will happen? Only the non-recombinants will grow because they are offering resistance. So only those ones will grow. So we will have two types of plates. Okay. Give me a second to copy paste this. Okay. So we have the plates here. Now this is the original plate and in this plate now we have added tetracycline. Okay. So the recombinants one here the tetracycline is broken, whereas the non-recombinants we know that the tetracycline resistance gene is intact. So here what will happen is only the non-recombinant cells will grow because they are still having the tetracycline resistance gene intact. So only those ones will grow. So what we can do now is we can easily select, okay, or we can pick up the recombinant from the master plate or the original plate, and then we can grow them separately. Okay, so these we can pick up and we can grow them separately, so that we will have a culture of only the recombinant transformer. So this is how we can use replica plating for selection of the recombinants from the non-recombinants. So all these things, whatever I am talking, they could be asked as individual questions also, or they could be asked as a whole, a question as in like a big question. Okay, so for this, it's very easy, though it is confusing, but it is very easy, because if you make some logic out of it, it will be very, very easy. Okay. So what would be the nature of the recombinants? They will be ampicillin resistant, but they will be tetracycline sensitive. So this we have to keep in mind. Okay. Alright. So we have finished one part, that is replica plating. So now let us deal with insertional inactivation in bit details, because this could be yet another question that would be asked for your exams. Okay. Now insertional inactivation. So we already know the concept of insertional inactivation. So let us take one example of yet another plasmid, that is PUC 18. We shall discuss more about the selection of transformants and non-transformants using PUC 18 plasmid in our part 2. So till then, keep studying, keep recalling, and like, subscribe and follow our channel for more details. Thank you.